Folding and assembly of biological macromolecules refer to the processes by which large molecules, such as proteins and nucleic acids, achieve their functional three-dimensional structures. The proper folding and assembly of these macromolecules are crucial for their biological activities, including enzymatic catalysis, molecular recognition, and cellular signaling. In this note, we will explore the key concepts and mechanisms involved in the folding and assembly of biological macromolecules. Proteins are composed of linear chains of amino acids, and their folding involves a hierarchical process that progresses from the primary structure to the tertiary structure. The primary structure refers to the specific sequence of amino acids in the protein. The folding process begins with the formation of secondary structures, such as alpha helices and beta sheets, which are stabilized by hydrogen bonding within the polypeptide backbone. The folding then proceeds to the tertiary structure, where the protein adopts its native conformation through interactions such as hydrophobic interactions, hydrogen bonds, and disulfide bridges. In some cases, proteins can further assemble into quaternary structures through protein-protein interactions, forming functional complexes. Here you can see the native and denatured state of the biological macromolecules. So pause the video for a while and read this slide.
Figure below shows some of the incredible assemblies that biology has invented using molecular interactions.
So pause the video for a while and read this slide. 2. Nucleic acid folding. Nucleic acids, including DNA and RNA, also undergo folding processes that are critical for their functionality. DNA adopts a double helix structure, in which two complementary strands are held together by hydrogen bonding between the nucleotide bases, adenine thymine and guanine cytosine. DNA can further fold and twist into higher-order structures known as supercoils. RNA molecules can form complex secondary structures through base pairing interactions within a single RNA molecule. These secondary structures, including stem loops, bulges, and internal loops, are important for RNA stability and function. Additionally, certain RNA molecules, such as riboswitches, can fold into specific three-dimensional structures that act as regulatory elements. 3. Assembly of complexes. Biological processes often involve the assembly of macromolecules into larger complexes. Protein-protein interactions play a crucial role in forming these complexes, where different proteins come together to carry out specific functions. These interactions can be mediated by non-covalent forces, including electrostatic interactions, van der Waals forces, and hydrophobic interactions. Similarly, proteins can interact with nucleic acids, such as DNA or RNA, to form protein DNA or protein RNA complexes. These interactions are vital for processes like gene regulation, DNA replication, and protein synthesis. In the case of membrane proteins, assembly involves their integration into lipid bilayers, often aided by chaperones and quality control mechanisms. IV. Factors influencing folding and assembly. Several factors influence the folding and assembly of biological macromolecules. Chaperones are specialized proteins that assist in the proper folding of other proteins. They can prevent misfolding and aggregation, facilitating the attainment of the native conformation. Post-translational modifications, such as phosphorylation, glycosylation, and acetylation, can affect protein folding and function. Environmental factors, including temperature, pH, and ionic strength, can also influence folding and assembly processes. Furthermore, molecular crowding, which occurs in the crowded cellular environment, can impact the folding and assembly of macromolecules. Techniques for studying folding and assembly. Several experimental techniques are employed to study the folding and assembly of biological macromolecules. X-ray crystallography allows the determination of protein and nucleic acid structures at atomic resolution by analyzing the diffraction patterns of X-rays passed through crystal samples. Nuclear magnetic resonance (NMR) spectroscopy provides information about the structure and dynamics of macromolecules in solution. Cryo-electron microscopy (cryo-EM) enables the visualization of macromolecular complexes by reconstructing three-dimensional images from electron micrographs of frozen samples. Forster resonance energy transfer (FRET) measures distances and conformational changes within macromolecules. Single-molecule techniques, such as single-molecule fluorescence microscopy, can provide insights into folding pathways and dynamics at the individual molecule level. In conclusion, the folding and assembly of biological macromolecules are complex processes that are essential for their proper functioning. Understanding these processes and the factors that influence them is crucial for elucidating biological mechanisms and designing therapeutic interventions. Ongoing research continues to advance our knowledge of these fundamental processes and their significance in various biological contexts.